So welcome to Mohit Agriculture Tutorials. My name is Mohit Gupta and today we have came with another interesting and new video on the topic cell signaling and signal transduction communication between cells, right? So in this video we are going to talk about what is cell signaling, how does it work, what are all the theories about cell signaling, and how does cell signaling influence the life of human being or any individual who is living, whether it is a plant, it is an animal, or anything which is living. We say it or which, then the thing is considered living. If the thing is considered living, signaling is working inside the body of any we can say living being, right? So over here we designate this topic to be important, as we have talked about ERGIC complex and endoplasmic reticulum Golgi complex. As important those topics were, the same importance would be given to this topic also, because this topic creates the pattern of what we say is transport of molecules from one place to another place, and whatsoever you see, whether it is endocrine functioning, hormonal synthesis, or anything whatever inside our body is being coordinated by signaling molecules, peptides and proteins going on inside our cell, and various diffusing inside our cell to various ligand and receptor binding that we see ever and ever in our society, in our neighborhood, or anywhere in every animal you see it. As you see, a child is small, then it grows. Literally we can say that signaling molecules are working inside the body of a child that performs certain task, or we can say that just we can say coordinates, or we can say what they just we can say that they have promoted those molecules that are being designated for that are the receptor or ligands for the we can say growth of a children. So in this way you can just think about signaling molecule diffusing in the cytoplasm of any cell that creates changes to our body, to our environment, to our neighborhood. Let's see what are the changes to our body, to our environment, and what is the cell signaling, how does we perform or anybody performs cell signaling, signal transduction communication between cells, right? Okay, so here in this image shows a Hawaiian bobtail squid, right? So what you see herein is that the squid is just, we can say literally it is lightening a lot. So this is particularly, we can say this is a symbiotic microorganism living in the body of the cell, body of the squid, that is creating somewhat lightning, that is just a type of, we can say, a trap by the squid that creates a trap, we can say just we can say attracting its prey towards itself, because it is a predator, and we know what predator is. Predator just hunts its prey. So the prey in the water is just moving across the water towards light, that we have already seen in microorganism also. They are also phototropic, so these prey are also somewhat phototropic and they move toward light. So the fish or any, we can say, organism living inside the body, inside the ocean, or any where by the way it is found perfectly in Pacific Ocean only. So any of the organism that is living inside the Pacific Ocean would be, we can say, attracted towards this squid due to the signaling molecules that are being designated in the cell due to the symbiotic relationship with bacterium or any microorganism which causes this we can say symbiosis causes this signaling pattern in the body of a squid. So the organism that we call bacteria vibrio fischeri is an organism that actually causes the signaling pattern like this, that lightens the body of the squid. Now how does signaling take place, and who signals these molecule? Is the squid lightning? No. This lightning pattern is seen among the microorganisms that are in the body of this, in the, we can say, on the body, sitting on the body of this squid. There is symbiotic relationship somewhat. They just help it to get its prey, and this is somewhat host to the vibrio fischeri. So what we see herein, that vibrio fischeri is just creating some light effects on the body of this squid and creating signaling, and somewhat signals are being created from one cell to another cell, because this also we tend to know that signaling is caused from cell to cell, right? So we can know about vibro fishery, that is a prokaryote. So prokaryotes always does signaling, but in signaling in the means of to attract another cell towards it, or somewhat we can say they are having signaling molecules. So these signaling molecules tend to attract the prey of the predator, right? So the thing that arises is that how does this signaling happen actually? What are the macro micro molecules that are working inside the body that creates such a signaling, such a we can say what very interesting topic this is. So such a we can say extreme level of signaling you can see here in light production is somewhat process that uses such a large amount of ATP, so a bunch of cells would be working over that position. So what are the macro micro molecule, and what are the molecular perspective in which we'll learn this signaling, right? So we'll know about it. Okay, so signaling is being done in three of the major ways. You can see here in autocrine, right, in which we can say it is a paracrine type of signaling, wherein this is a receptor, this is a ligand, and this ligand approximately binds to the same receptor on the same cell on which it is designed to, right? So it is designed for the same cell which creates this ligand, and this ligand you can see from the originating from the lumen of the cell to the receptor, which is on the similar cell. So same cell when creating signals, so this type of signal is called paracrine signal. Then we have endocrine signal, or where you can see here in we can see what the endocrine signal is seen here, wherein the blood is flowing, and flowing blood is carrying some we can say what molecules to places, right? Here you can see paracrine signaling. This is autocrine signaling, right? You can see here in paracrine signaling from one cell to the another cell. So signaling molecules are diffusing in the cytosol in the similar manner. They just bind to a receptor. There is a receptor, there is a ligand which binds to the receptor, creates some signaling, we can say proteins activated inside the lumen of the cell or anywhere in the cytosol. Once cytosolic ingredients are activated, that may create some changes in the environment or anywhere which cell want to, right? So very first is autocrine, in which similar cell is targeted by the ligand. The second one is paracrine, in which the different cell are targeted, and the last is endocrine, in which blood stream is used to transfer the sensory molecules which are activated due to the binding of ligand and receptor, right? Okay, so short review about what signaling is, how does it is created. So you can see here in this is a signaling cell. We are showing the oval plasma membrane. Plasma membrane is designed with certain receptors. You can see this cup type of body as a receptor. There are certain ligands. You can see here in the balls on the cup. These are ligands that create the effector protein that we can say transfers the information to the effector protein. Once effector protein is targeted to the another receptor, then second messenger is activated. In the similar manner, third messenger and fourth messenger in the similar manner, messengers are activated. We can say what series of activation is there, in which various type of second, then tertiary, quaternary. In the same manner, there are various signals to be activated to create a whole we can say effect in the cell, right? So this is a particular we can say what's transcription survival of protein synthesis. This is the thing to remember is that these signals that are always created in the cell ends with transcription and translation. So inside the nucleus, DNA is the king, as I have already told you, of the cell, and DNA tend to work like that it has to create some proteins. So these all signaling molecules, when bound with receptors, will create changes in the cytosol that would lead to the translation and transcription of the protein. That's it, right? Okay, so herein that you can see is that we are having a protein kinase. This protein kinase got activated by some signaling molecule. It phosphorylates protein kinase 2, then protein kinase 3 phosphorylates protein kinase 3 again, and transcription factor once associated with these kinases got phosphorylated, and CAMP binding and all those things that are signaling molecules going inside the cell, that we can say what, that signaling molecule are designed to create some function, right? So functions are created inside the lumen of, we can say, nucleus, wherein the DNA is just transcribed and translated. So you can see herein the transcription of DNA is being carried out by kinases, and kinases has particular, as we have told earlier also in molecular biology, that kinases create certain functions in the, we can say, proteins that are being phosphorylated by kinase. So phosphorylation, or we can say PPI function of these kinases creates some designated function to the DNA via polymerase that creates mRNA, right? So this is a fundamental law of signaling, that once a receptor bound to the ligand, it would create, we can say, effect where in the nucleus, right? Once in the nucleus, this signal has arrived or has reached to, right, say calcium is being generated by endoplasmic reticulum by binding of IP3 protein. Say what, I am giving just an example, we will talk about later what is IP3 protein. So once IP3 protein generates some messages, this would lead to phosphorylation of calcium molecules, means calcium will bound to calmodulin, and calmodulin will create some effects that would lead to DNA to be transcribed and translated, right? Okay, so this would be the end of this video, because this must be the introductory video only, because long videos I don't think so that you can see the whole process in this single video only. We will make another video, right? So in this diagram we are showing somewhat a perspective how does ligand and receptor bind. So herein you can see receptor is there in the wall of plasma membrane, wherein you see this alpha beta gamma unit. Now the main question arises, you do not see here in gamma, you only see here in alpha. So the main question arises, what are these alpha beta gamma units, what are these alpha beta units, and how does they create function inside the cell? So herein if you see, once ligand bound to the receptor, say this is the receptor, you can see here in 3D diagram, so this is a receptor. Once ligand bound, say this is a ligand, so it creates function. What type of function it creates? Its target is G protein, so it activates this G protein, that is alpha beta unit, merge all together. So this G protein once activated creates some function, that was what I would say that they will just associate with this cis retinal, we can say, rhodopsin molecule. This is a rhodopsin that is inactive. Once G protein is associated with this rhodopsin molecule, would create proteins that would cause function in the cell. So functions in the cell are created by the membrane bound machinery, right? Okay guys, so next video would be on G proteins, right? We'll talk about how does G protein work inside the cell, how is signal transduced inside the cell via G protein, GPCRs, right? What are various GPCRs, and how does the receptor and ligand binding takes place, how does GPCR associates with the receptor complex, and how does G protein, the alpha unit disassociates with the receptor complex, and once disassociated, how does that dissociation would lead to, we can say, what the another we can say secondary protein to be activated, effector protein, and once effector is activated, how does that what else processes are carried out inside the cell. We will talk about that. So in this video, this was just an introductory video, what cell signaling is and how does the cell signaling work inside the cell. So if you like this video, please subscribe, hit the bell icon. Thank you. Jai Hind, Jai Bharat. Thank you.